Hey, hey, I was thinking about you last weekend and
Okay.
I realized I don't even know your name.
Look, I like this seat. I really do. It's not too far that I can see and not too close that it's awkward with Leon. I mean, the professor. If we could just keep this here more quiet, that'd be great.
Oh, I get it.
Sorry, I didn't mean to be harsh. It's just I'm here to attend the class and come back, and that's it.
No, I get it. You must be one of those, how do they call them?
What do you mean?
Kind of weird.
Thanks?
An introvert.
Last
Seriously?
time we discussed the early efforts behind the quantitative study of perception. It seems that it was in fact the University of Leipzig, where the work of Weber, Fechner, and Wundt took place, that was the true founding figure of experimental psychology. It was a theater of the ultimate divorce between psychology and philosophy, when the former truly started to standardize its methodology. Although the psychophysics still relied heavily on their subject retrospection as the information's point of access. This approach was prone to criticism because many scientists, appropriately, thought it might filter, if not completely distort, the perceptual experiences. The debate regarding the importance of qualitative data to study human psychology is not illegitimate now, and it certainly was not then, and resulted in a highly fractured field, with different schools disagreeing on either its methodology, its subject, or both. This fracture would prevail for some time, until the steady neuroscientific advances of the 20th century culminated in a shiny new method for collecting information. Being able to study human psychology without relying on manifest behaviors really was a revolution for the field. So, once more, let's rewind the tape to witness the fascinating story of psychology, this time from the point of view of neuroscience. We are still in the aftermath of the Renaissance, with the emperor's zeitgeist serving as the backdrop of many great methodological advances. Undeniably, the days of approximate surgeries and spiritual theories of the Middle Ages had long been left behind when Charles Bell, a British physiologist, made a huge contribution to the study of the nervous system. By dissecting rabbits, he was the first to issue experimental evidence for two functionally distinct types of nerve, the sensorial and the motor. He discovered that sensory nerves forwarded impulses from the sense receptors to the brain, while motor nerves carried them from the brain to the muscles and glands. There had been theories that the brain hosted distinct areas for distinct functions, but now there was scientific proof that specific mental functions were mediated by different anatomical structures. Fast forward a few decades, German physiologist Johannes Müller demonstrated there was in fact five types of sensory nerves, each resulting in characteristic sensations when stimulated. The visual nerves prompted visual sensations, auditory nerves, auditive sensations. I'll let you finish that sentence. Uh, Surprisingly enough, though, the nature of the stimulation was not important, meaning that stimulating the visual nerves with electricity, pressure, or by slipping on a pair of socks and falling on the head would all cause seeing stars, no telescope required. That did not mean, however, that the nerve sensitivities were all equal, prompting, as you probably know, a finer visual response to actual light waves. When Mueller discriminated 12 pairs of cranial nerves, he became unsure if the different sensitivities of the five types of sensorial nerves emerged from their intrinsic properties or just from the area of the brain they're wired into. We now know the latter is true, that the nerves themselves are similar to each other, while the sense organs, including the brain area that supports them, differ in what type of stimulation they are acute to. Further evidence for a localization of faculties came from clinical data. For example, in 1861, a French physician named Paul Broca was able to show experimentally the localization of speech in the brain. He had been called to give care to a man with a gangrenous infection at the Hôpital Bicêtre in Paris. Once there, he realized that the poor soul had had his whole life a bizarre speech impairment where he could only repeat the sound tan. If he wanted to ask for direction, he could say tan 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 tan. If he knocked his little toe in the dark, he would say tan 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 tan. But he seemed otherwise perfectly healthy. He had a normal comprehension of language and could communicate intelligently by signs. Broca was unfortunately unable to save his new patient's life. However, his autopsy granted him a clear demonstration of how the atrophy of a particular region in the left side of the brain was associated with the verbal production of language. Of course, this did not elucidate whether the neurological damage caused the functional deficiencies, or if it was the other way around, the functional deficiencies prompting neural atrophy. But the association between a localized brain area and a well defined cognitive function was now indisputable. Further localized atrophies, such as those caused by bullet injuries during the early 20th century wars, permitted other neurological associations with specific functional troubles. Around the same time, new technological advances allowed the precise electric stimulation of brain regions in patients undergoing brain surgery, usually to remove epileptic foci. This led neurosurgeon Wilder Penfield to create interesting representations of what a man would look like if his body part sizes were proportional to the brain areas responsible for those functions. He made one cortex man for both the sensorial and motor system, probably to show that size does not matter, it's how much your brain is dedicated to it that does. As the progress of technology, fueling methodological improvement, was slowly taking place, the theoretical context faced an important shift when Darwin published his Origin of Species and The Descent of Man between 1859 and 1871. Even though some unfortunate circumstances had made possible important scientific discoveries, a more systematic study of physiology would only be made possible once Darwin would provide an explanation for the mechanisms of biological evolution. In demonstrating that humans inherited from the ape by a continuity in his bone and organ structures, it followed that understanding the animal's brain could teach us many things about humans' brain. In other words, evolution made possible the investigation of the animal's nervous system as proxy for humans. This would have tremendous consequences for the field of neuropsychology, but also for the rest of psychology, notably with the behaviorists and functionalists. That's because understanding the biological evolution can serve as a framework to guide, but also to ground the realm of possible theories. For instance, you are probably aware of the theory that humans typically use only 10% of their faculties. However, the evolutionary framework contextualizes how the persistent passing of unutilized brain matter through generations is impossible, as it does not confer any adaptive fitness and would, on the contrary, prove wasteful. That's not to mention that simple functional analysis show that every area of the brain can be activated by rather mundane tasks, and always in large aggregations, but that's beside the point. We now seem to know quite a lot about the brain. We know it's a 3 pound network of some 80 billion neurons, and that each of these have hundreds, if not thousands, of connections with others. We know the cerebral cortex is a 2 mm thick layer that hosts the cognitive functions. The primary receiving area for vision occupies most of the occipital lobe. The area for hearing is located in the temporal lobe. The parietal lobe hosts the skin senses such as touch, temperature, and pain. As we'll see for every sense, once a sensorial signal reaches the corresponding primary receiving area, it is quickly transmitted to many other structures in the brain. For example, the frontal lobe receives signals from all the senses and can thus integrate that information and compare to what is in memory to achieve categorization. The accessibility of knowledge today makes it hard to appreciate the amount of research that had to be done in order to construct it in the first place. Of course, what I've included in the first part of this lecture, The discovery of sensory nerves, the experimental proof on the localization of functions, and the conception of an evolutionary framework does not even come close to grant justice to all the incredible neurological discoveries of the last few centuries. The reason for these shortcomings is that I wanted to cover the functioning of a few neuroimaging techniques that uncover most of the knowledge you'll earn in this course, but also to give a crash course on how neurons work. The good news is that we're going to have not one, but two breaks today. Hooray!